Bismillahirrahmanirrahim. Today we are going to discuss the action potential. My name is Dr. Kachiwal. I'm professor and chairman of the department of Physiology and Agricultural Institution. An impulse is initiated by stimulation of a sensory nerve endings or by the passage of an impulse from another nerve. Transmission of the impulse or action potential is due to a movement of ions across the nerve membrane. There are two types of the neurons. One is myelinated neuron and non-myelinated neuron. Myelinated sheath has to cover the axon is the that allows the flow of impulses through the axon to the terminal extent. So the neurons is consist of a cell body, dendrites, nucleus, axon, and the synapse. So you can see that the special properties of the neuron is that it causes excitability, action potential in axon, conduction means action potential in axons, then transmission, synapses and electrical and chemical transmission, integration, postsynaptic cell, plasticity, that presynaptic terminal and postsynaptic membrane. So we can see that the typical neuron is having the body parts like a dendrite, soma means a cell body, nucleus, and then axon. Then lastly, it attaches with the motor neuron is attached to the muscles and develops the synapse over there to transmit the message from the sensory neuron to a motor neuron, and then the motor neuron transmit the impulse from through axon to the muscles, and the message is reached to the muscles to contract or relax itself. So this is the one of the example that the nerve impulse. A nerve impulse is the electrical message. It's not a current, but it's an electrical message that is transmitted down the axon of a neuron. Then the impulse does not travel directly down the axon, but is generated at a points along the axon. Means the impulse or the action potential is developed along the axon. So there is a the resting potential, and then the action potential, then the resting potential, again the action potential. So likewise, the action potential or one impulse is in between the two resting potentials. So the speed of nerve impulse ranges from approximately 1 mile per second to 100 miles per second. So if you see the resting membrane potential, the resting potential of a neuron refers to the state of a neuron prior to the sending of a nerve impulse. Nerve impulse से पहले जो condition होती है nerve की, उसे resting membrane potential बोलते हैं. At this point, the neuron is said to be polarized. और इस condition में neuron जो है वो polarized होता है. एक साइड पे नेगेटिव चार्ज होती है और दूसरी साइड पे पॉजिटिव चार्ज होती है, जिसकी वजह से हम उसको पोलराइज्ड न्यूरॉन या रेस्टिंग मेम्ब्रेन पोटेंशियल बोलते हैं, जिसमें -70 मिलीवोल्ट्स डेवलप होता है चार्ज और इलेक्ट्रिकल जो है वो एक्टिविटी. An impulse is initiated by stimulation of a sensory nerve endings. Sensory nerve endings जो है वो मोटर न्यूरॉन को ट्रांसमिट करती हैं impulses, और transmission of the impulse is due to the movement of ion across the नर्व सेल मेम्ब्रेन। और ये जो एक्शन पोटेंशियल है डेवलप होती है या इम्पल्स डेवलप होती है व्हेन द आयंस और सोडियम आयंस क्रॉसेस द cell membrane and depolarize the cell, and it is reaches to the peak whenever it generates plus 50, plus 30 or 35 millivolts, and then one peak is developed, one impulse is developed, and then again repolarization, the potassium from the potassium channels go out, and repolarization takes place, which has to last the millivolts which were developed, and it comes towards the minus 70 millivolts, which is a resting membrane position. So this means that there is a different electrical charge on each side of the membrane, which is called a resting membrane. Resting membrane में जो nerve cell के बाहर चार्ज होती है आयंस की, वो पॉजिटिव होती है, और इन जो होती है वो नेगेटिव होती है, और इसलिए बोथ साइड्स आर बीइंग पोलराइज्ड, एंड देयरफॉर दिस polarized condition is known as a resting potential of the nerve. The action potential, when the nerve produces the impulse, when a nerve impulse is transmitted along a neuron, a wave of electrical activity passes it along the axon, not a flow of electrons. So it is not an electrical current. It is the activity which is electrical activity which passes along its axon. This can be detected by using the cathode ray oscilloscope with the electrode placed inside and outside of the neuron as a transient change in electrical change on the membrane surface. This is called action potential and is the basis of nerve impulse. And in the action potential, the in the resting position, the mm, the activity is minus 70 millivolts, and when it is initiated by the stimulation of a sensory nerve endings, the threshold is reached at a 55 millivolts, and then another sodium channels opens and then give the passage to the sodium, which causes an inflow inside of the membrane. Therefore, this depolarization develops the millivolts, there's a 35 millivolts on which the peak develops, and then it is by a repolarization of the potassium, it comes down towards the base of a resting position, and reaching before the resting position, the hypopolarization is being shoot up. So this undershoot is known as a refractory period, and then the resting state develops. So in this graph, we can see that the minus 70 is the resting position, threshold is minus 55 millivolts, and then the action potential reaches at 35 millivolts, and then repolarization takes place, potassium comes out, and then it is causing a hyperpolarization, hyper depolarization. So it goes beyond the resting position, which is known as a refractory period, and then comes to a resting position, which is minus 70 millivolts, and then it remain in this way. So you can see that the action potential developed from a minus 70 millivolts reaches to a 35 millivolts, and then come down to a refractory position, and then the resting position, and this is known as the resting membrane potential, threshold, an action potential, and then again refractory period, and then the resting position. So this is the one of the chart where I'm showing you the resting potential is a period on which the now remain polarized at a minus 70 millivolts, and then the stimulation causes to rise up and cross, you open the sodium channels cat, and then the sodium enters into the cell membrane inside, and then it rises the millivolts, which may have to reach up to a 35 to 40 millivolts, and this flood of the sodium causes the generation of the ion activity, the electrical activity, and then after reaching at a peak, it reverses back by opening the potassium channels, which gives passage to uh, the potassium, and then hyperpolarization is developed, which has the period which is known as the refractory period due to the active pumping of the sodium and the potassium during the refractory period. And then the threshold has come up, and this has to develop the one resting potential, then the action potential, then the resting potential. So in two resting potential, one action potential is developed. So the action potential is a resting potential. The sodium and the potassium special voltage gated ion channels closed, and neuron is polarized. So the sodium gated channels, when the protein opens due to the change in the shape of a protein itself, makes the membrane more permeable to a sodium ion. Now, which allows the sodium ions to diffuse into the neurons down the electrical chemical diffusion gradients. This causes the inside of the neurons to become increasingly more positive ions, positive volts. So this is the depolarization, and neuron has become depolarized after reaching at the peak where the 35 or 40 millivolts are developed. This is known as an action potential peak. Whereas when the action potential after a stimulation reaches at the 55 millivolts, that is known as a threshold, and this is the minimum level of the stimulus necessary to cause a depolarization. Then the further the annals and the sodium channels opens, and then the flood of the sodium go inside and causes the peak of the action potential, and are all or nothing. The all action potential are the same size irrespective of the intensity of the stimulus. So consequently, information about the intensity of the stimulus is coded in the number of impulse, a strong impulse, many action potentials. So the properties of the action potential have a refractory period about a 1 millisecond. The ion channels remain closed and cannot be made to reopen, so no further depolarization is possible. The consequence are it separates the impulses from one another by a fraction of a millisecond, and so imposes a maximum on the number of impulses which can be transmitted along a neuron in a given time. It ensures the impulses can only pass in a one direction along an axon forwarded into the next resting region, but not back into a region still in its refractory period. Role of the action potential in the transmission of a nerve impulse. A stimulus causes the sodium-gated channels proteins to open, effectively causes a change in the shape of the protein itself, which allows the sodium ions to diffuse down the concentration gradient, cross the membrane into the cell, and that so it sits so set off in action potential. Nernst equation E ion is equal to 2.303 RT divided by ZF log of ions and divided by ion in. Ion out log of ion out divided by ion in. So E ions means the ionic equilibrium potential is equal to Z means charge of the ion, F means a Faraday constant, T means absolute temperature, R means universal gas constant. Next equation relates the voltage across the membrane, which is in equilibrium with the concentration of ions inside and outside the cell membrane. So the ionic distribution in the inside the potassium is 125, whereas the sodium is a 12, chlorine is a 5, water is a 55,000, and anion is 108. Outside the position of the ions, the potassium 5, sodium 120, chlorine 125, and water 55, and 0 anions outside. So therefore, the potassium can cross the permeability. Sodium cannot cross the permeability. Chlorine can cross the permeability, and water can cross the permeability. So synaptic transmission, first of all, the stimulus will affect on the presynaptic neuron that causes the depolarization, which develops the impulse, and then the presynaptic membrane that opens the calcium ion entry, and synaptic knob will have affinity of a synaptic vesicle to bind with a presynaptic membrane, which release the neurotransmitter via exocytosis, and synaptic cleft binds to a synaptic cleft and reach to a specific receptor of a postsynaptic neuron. So the postsynaptic neuron will excitatory neurotransmitter allow the permeability to a sodium, a repolarization takes place, and excitatory postsynaptic potential. So inhibitory neurotransmitter will open the potassium and chlorine channels and causes the hyperpolarization, and inhibitory postsynaptic potential will develop. So conclusion is that the nervous system detects and responds to a changes inside and outside of the body. Together with the endocrine system, it controls many vital aspects of the body functions. The nervous system consists of the brain, spinal cord, peripheral nerves. The structure and the organization of the tissues that forms these components enables the rapid communication between different parts of the body. So the propagation of the nerve impulse that starts from the speed of a conduction of a nerve impulse, the transmission speed can range from 0.5 um, miles per second to over 100 miles per second. Impulse transmission can be speeded up by increasing the diameter of the axon. So this the diameter of the axon is a jump. It will produce more speed and causes the neuron to reach at a destination with a higher speed. Neuron having a myelin sheath. This is rich in a lipid called a myelin, which makes the axon impermeable to ions, so action potential cannot be generated by the myelinated regions. So it acts as an insulator. So the action potential can only be generated at the nodes of rain wire, solitary conduction. So the local currents involved in nerve impulse transmission flow over a longer distance. Thus, the action potential seems to jump from node to node. This is called salutary conduction. Temperature effects on a nerve impulse, which affects the rate of diffusion and the rate of the energy release by respiration for active transport. Since it is controlled by the enzyme, the consequences is that the nerve impulse transmission faster in the endothermic animals, which maintain a high body temperature, and the synapses is also called a neural junction, the site of a transmission of electrical nerve impulse between the two nerve cells. And this is not a physical junction. This is actually a small gap of approximately 20 nanometers between the cells. So there is no membrane continuity. So the nerve impulse can not be crossed directly. Transmission of a impulse through synapses. Instead, the transmission is chemical called neurotransmitters. These made in the Golgi body, and synthesis requires energy from respiration, is stored in the vesicles in the synaptic club, synaptic bulb. And there are several different types of the neurotransmitter. For example, acetylcholine used in the voluntarily motorized neurons, muscles, and parasympathetic nervous system, whereas the noradrenaline used in the sympathetic nervous system. So action potential um, and action potential arrives when the membrane depolarizes calcium ion channels, open the calcium ion, enters the neuron. Calcium ions cause a synaptic vesicle containing a neurotransmitter to fuse with the presynaptic membrane. Neurotransmitter is released into the synaptic cleft that the neurotransmitter binds with the receptors on the postsynaptic membrane. Cations channels open the sodium ions for through the channels. So when released, the neurotransmitter will be taken up across the perisynaptic membrane whole or after being broken down, or it can diffuse away and be broken down. So the membrane depolarizes and initiates an action potential. You can see the axon and the synaptic vesicle, neurotransmitter in the synaptic vesicle, then the perisynaptic membrane, and the uh, synaptic vesicle burst releases the uh, so the acetylcholine and reaches to the postsynaptic membrane where the sodium channels open and gives the passage to sodium. So type of a synapse, one is the excitatory synapses, binding the neurotransmitter to postsynaptic neuron, opens the sodium gated channels, and then the sodium diffuses in and depolarization takes place. Action potential develops, and so the nerve impulse can continue around the nerve circuit. Whereas the inhibitory synapses that binding the neurotransmitter to a post synaptic neuron opens the potassium gated channels that causes diffuses the potassium out, and inside the neuron becomes even a more negative and so impossible to depolarize. No action potential develops, and so the nerve impulses cannot continue around the nerve circuit. Neurotransmitters, major categories of the neurotransmitter, including the amino acid, glutamine or GABA, the acetylcholine, uh, monoamines like serotonin, dopamine, norepinephrine, epinephrine, then the purines, adenosine, and the gases of nitric oxide. Thank you very much. So these were the transmitters, and I hope that you will share this lecture to your friends so that they may have to prepare themselves And I hope that you will even subscribe the channel so that we may have to reach the majority of the peoples, and you may have to receive the coming lectures on your internet. Thank you very much. Thank you.